So, something we're a little bit differently today. Ooh, sorry, brain left for a second. I'm gonna see how slideshows work for us. Just through probably the beginning of genetics. If it's something that we decide we don't like, I'll go back to writing it out. But if it's something you'll like better, we can keep it around for a little bit. So, yeah, you can turn in the packet whenever. Just chuck it on this desk right here, and I'll get you. So, did anybody have any questions or issues with the Punnett squares yesterday? No? Okay. Y'all had worked with monohybrid and dihybrid crosses on Punnett with Punnett squares before? Yeah, no, like that is exactly what this was meant to do. Just kind of jog your memory a little bit. And we're definitely not done with Punnett squares things, but today is all about how we can use probability to figure out the results of a Punnett square without actually having to do a Punnett square. So instead of actually having to like write out the Punnett square and figure out everything and then just count our results, we're going to use a little bit of math this morning to kind of bypass it and see what happens. So we're going to talk specifically about the multiplication rule and the addition rule of probability, and we are going to predict genetic outcomes without the use of a Punnett square. How's that sound? Okay. So that is really small. Let's not do that again. So I'm going to have to fix a bunch of these. That's the one thing with slides is I don't know how big to make the font. Ta-da. Okay. So when we talk about probability, the scale ranges from 0 to 1. So something that is definitely going to happen, that help, or that, those are the exact same thing. So probability ranges from 0 to 1. Something that is certain to happen has a probability of 1. Something that is certain to never happen has a probability of 0. So if I flip a coin, right, the probability that it will be heads or tails is going to be 1, but the probability that it will be purple is 0. In a coin toss, the chance of heads is 1/2. In a deck of cards, the probability of pulling like the ace of spades is 1 out of 52 because there are 52 cards in a deck. We know this for sure because all of these are independent events. If I were to flip one coin with one hand and one coin with the other hand, the outcome of one will not affect the other one. So, this means that it is an independent event, just like our genes segregate into gametes, right? The probability that one Mendelian gene will go into one gamete is it does not affect the likelihood of another gene being segregated into the same or different gamete. This independent event thing only applies to genes that are inherited through Mendelian stuff. So Mendel got lucky when he chose pea plants because all of the characteristics that he chose segregate independently of one another. And humans, right? Our inheritance is going to be a little bit different. There's a little bit more sway one way or the other. There are some traits, though, that are inherited through Mendelian genetics. If you have a widow's peak, right, if your hair goes into a point down in the front, that is inherited through Mendelian genetics. If your earlobes are attached or free from the side of your face, that is a Mendelian trait. So we can track that stuff through these probability rules, but not something like height or eye color because those are influenced by multiple traits, which we'll talk about next week. this week. End of this week. Fun times. So, independent events, like stuff like this, can be determined through probability laws, not just Punnett squares. So yeah. Any questions so far? Reasonable? Okay. That's the one thing I gotta work on with doing this is pace, because I tend to talk really, really quickly. So if I ever get going too fast or if I move too fast, just yell at me. I'll go back. It's a little bit of a learning curve. Cool. Yes, being able to paraphrase what is on a slide into your notes is a skill that will definitely come in handy. I had my first ever biology professor in college. Didn't use PowerPoints. Didn't just like talk. What he did is he had. Do you guys remember the old timey projectors with like the clear plastic sheets? Right. He had one of those on a roller, and he would write and he would roll it as he wrote, and we just had to keep up. I had my math professor would do practice problems and would have a doc cam and just a stack of printer paper and a Sharpie, and would just do practice problems and throw the paper and do practice problems and throw the paper. So being able to paraphrase is a very important skill. So the multiplication rule. That's where we're going to start. So the multiplication rule states that to determine the probability that two or more independent events will occur together in a specific combination, we multiply the probability of one event by the probability of another event. So if we want to know the probability that an offspring is, we'll say, big R, big R. We want to know what the likelihood that we are going to have two dominant alleles in an offspring, right? We could do our Punnett square. Big R, little r, big R, little r. Right? And we can see that only one of our four offsprings has both dominant traits. So we could say that that is 1/4. And in simple monohybrid crosses, it might just be easier to do the Punnett square really quick. But what you could do, so if this was a coin toss, right? Coin toss, right? So you flip it, heads is dominant, tails is recessive. We have a 50-50 shot. So the probability of getting the big R is 1/2, right? And the probability of getting a big R from this one is 1/2. So the probability of both of those happening together is 1/2 times 1/2. And I think I've got it written out on the next slide a little bit clearer. So 1/2 times 1/2 is 1/4. So we could do it mathematically without using the Punnett square, or we could do it with the Punnett square and we get the same answer. So while you're figuring stuff out, if you want to do it with probability and check your work with the Punnett square, that would work too, because you should theoretically get the same answers. That was a little messy. So the probability of two independent events occurring is equal to the multiplication of the two events. So, oh, no. Sorry, I can rewrite that out. Does anybody have any questions about the multiplication rule? No? Okay. I went way too far ahead. Come back. That's not what I want. Okay, there we go. The addition rule. right? This is just an example. This is our definition. The addition rule states that the probability of any two or more mutually exclusive events will occur is calculated by adding their individual probabilities. So let's say we want a heterozygote from this cross. We want an offspring with the same genetic material as their parents, right? So we could do the Punnett square, easy peasy. Right, and we can see that it's 2 out of the 4 or 1/2, right? That's one way to do it. That's the way that we default know how to do it. But you could also do it using the addition rule. So what you would do is you find the probability that you get the dominant allele from the egg and the recessive allele from the sperm, and that's 1/4. So the probability of getting this specific recessive allele and this specific dominant allele, right? We got a 50-50 shot of getting this one and a 50-50 shot of getting this one. 1/2 times 1/2 is 1/4, right? So you have a 1/4 shot of getting this recessive allele and this dominant allele. That makes sense? Okay? And then, but there's another way to get a heterozygote, and that is with the recessive allele from the egg and the dominant allele from the sperm. So there's a 1/2% chance of getting 1/2%. That doesn't make sense. We have a probability of 1/2 to get this dominant allele and a probability of 1/2 to get this recessive allele. So we have a 1/4 chance of getting this combination. So if we want to know what the likelihood of getting either of these, we can take 1/4 plus 1/4, and that will give us 1/2. Does that make sense? To where if we want the probability of just one of these occurring, we can multiply. But if you have multiple ways to get the same thing, then we can add. So with, I promise they'll get cleaner. With the monohybrid crosses, it might be easier just to fill out the Punnett square because that's what we know. But when we get to dihybrid crosses, it can get a little bit more complicated. So if we're looking at this specific dihybrid cross, right? So we've got a parent over here who's heterozygous. So big R, big Y, little y. And this parent over here is also big R, big Y, little y. That's our Punnett square we're going to kind of check our work with, just so you have it. So we can use the same rules of probability to predict the outcome of crosses with more than one characteristic. So dihybrid Punnett squares might be a little bit more complicated to do, might take a little bit more time in a time-sensitive situation, like an exam, cough, cough, wink, wink. It might be easier to go through and do it with a probability, and then at the very end, go back and check your work with the Punnett square if you've got the time. In something like a trihybrid cross with 3 traits, right? Definitely not going to want to do that Punnett square. So you can have the probability rules help you out there. So that's our example that we're going to work with. So I believe, yes. So the recessive trait so the R's are for round or wrinkled. So if you've got a dominant allele with the R's, that means we're going to have a round seed. If you have both the recessive, you're going to get the wrinkly seeds. And then Y is with color. So dominant allele is yellow, recessive allele is green. That's what we're going to be working with. So yes, okay. We're going to do specifically first a monohybrid cross with heterozygous yellow plants, right? So we do a Punnett square really, really quickly. 2 heterozygous, big Y, little y, big Y, little y. Okay. We know that the probability to get a heterozygous offspring is 1/4. A homozygous dominant offspring is 1/2, and a homozygous recessive offspring is 1/2. The exact same thing helps us with our R traits. Big R, little r, big R, little r. We have a 1/4 chance of getting a heterozygote, a 1/4 chance of getting a homozygous dominant, and a 1/4 chance of getting a homozygous recessive. These are our baseline probabilities. This is what we will be adding and multiplying to figure out what we're doing. Yeah? So this is our first step. Just figure out these baseline probabilities. Yes? I mixed them up in my head. No, you're good. Thanks for catching it. 1/2 probability getting a heterozygote, 1/4 getting a homozygote. So, with these probabilities, so those Punnett squares right there are the first 2 lines of this. With these probabilities, we can use the multiplication rule to determine the probability of each of the genotypes in the F2 generation. So if we wanted to know the probability of getting big Y, big Y, big R, we multiply the probability of getting big Y, which is 1/4, by the probability of getting big R, which is 1/4. So in our dihybrid cross, there's a 1 in 16th chance of getting 2 dominant Ys and 2 dominant Rs. Does that make sense? And if we were to go back to our Punnett square, right, we could make the dihybrid cross and then look through all of these and see that only one of them has both dominant ones. Or we can do the math really quick. Same thing, if we wanted to know the probability of getting a big Y, little y, big R offspring, we have a 1/2 chance of getting a heterozygote with our Y trait and a 1/4 chance of getting a homozygote dominant with our round trait. Multiply those together, there's a 1 in 8th chance of getting this outcome with the heterozygote for Y and homozygote dominant for R. So you can do this math, or you can go back and see that, okay, we want homozygote for y and double dominance for r. So we could look through them, do the dihybrid cross. You might think this is more, you might think this is simpler than doing the math, but the more comfortable you get with the math, the quicker this will be than doing this dihybrid cross. Any questions? Doing okay? All right. So if we want to know, I made this slide so I didn't have to keep clicking backwards. So like I said, big Y, big Y, big R, little r, or big R, 1 out of 16. This one right here. Big Y, little y, big R, here and here, 1/8, or 2 out of 16. Does that make sense? Okay. Any questions? Okay. All right. We had a little bit more of a complicated problem, and that is bringing our addition rule into it. So let's say we have a trihybrid cross, right? So we want to know, so we've got our Y's and our R's, but we want to bring pod size into it, or pod shape into it. So now we have added a heterozygote trait with P's. So now we have a heterozygote for this one, and for this one. But a heterozygote for our P's, and then homozygote recessive for our other traits. This is what we need for a trihybrid cross. You don't want to do it? Do a trihybrid cross? How do we even set it up? What's our first step? Yeah, we got to FOIL it. So you can start out with this one right here. So we can have big P, big Y, big R. We can have big P, big Y, little r. We can have big P, little y, big R. We can have big P, little y, little r. All the different combinations up top, all the different combinations down the bottom. We can have little p, big Y, big R, little p, little y, big R, little p, big Y, little r, and little p, little y, little r. Yeah. Do it down the side. It's a little simpler. We can have big P, little y, little r, big P, little y, little r. Big P, little y, little r. Big P, little y, little r. Little p, little y, little r. Little p, little y, little r. Little p, little r. So that's our setup alone. Can I make this smaller? I can. Now go away. Then you'd go through and you'd do your little combinations. So this one has big P, big Y, little r, big R, little r. And you do that for all of these. I mean, you can. You want the marker? Like fill one of these Punnett squares out? Not if you can do the math. That's why we're learning the math. So if you get stuck with the trihybrid cross, you could do the Punnett square, but you could also do the math. Last year when I was student teaching, I gave one of these as extra credit to my bio ones, and 2 people did it. So it's fun, but it just takes a lot of work. So that's a tri-hybrid cross. In order to not do these, we learn the probability rules. So I'll erase all our hard work. Go away. Please go away. Oh, close enough. All right. So, example problem. Using this exact same cross, we want to know how many offspring exhibit recessive phenotypes for at least 2 of the 3 traits. How many offspring will have recessive phenotypes for at least 2 of the 3 characteristics? Step 1, what are those genotypes? What genotypes will give us the wanted phenotypes, right? Because we want to know phenotype probabilities, not genotype probabilities. So we want to know what genotypes we could get, that give us at least 2 recessive phenotypes. So what's one? What's a genotype that gives us 2 recessive phenotypes? All right, so which one? Wait, you're gonna have to give me bigs and littles. Yeah, that's one. What's another one? Yep, that's another. Yeah. We want the genotypes, so the letters, that give us at least 2 of the 3 recessive phenotypes. That would give us one, but is that even possible with our parents? No. So yes, you could, but the probability would just be 0 because it's impossible. Yes, yes it is. Because we Yep, and there's one more. At least 2 of the 3. That's our step 1. We have 1, 2, 3, 4, 5 different combinations that will give us what we need. So that's our step 1. Step 2. Yep, okay. We were correct. Step 2 is to calculate the probability of each of these genotypes. So I did the first one for you, right? So the probability of getting little p is 1/4, same as over there, but with p's instead of y's and r's. The probability of getting little y is 1/2 because of the different crosses. We have different crosses. So I could have just left the Punnett square there. So we have big P, little p, big P, little p, big Y, little y, little y, little y, and big R, little r, little r, little r. These were our crosses, correct? Okay. So doing 3 little Punnett squares is still a lot easier than doing that big trihybrid cross at the beginning, yes? Okay. So with our next one, right? What's the probability of getting little p, little p? 1/4. What's the probability of getting big Y, little y? What's the probability of getting big R, little r? 1/2. So 1/4 times 1/2 times 1/2 equals 1 out of 16. What's the probability of getting big P, little p? Little y, little y? Little y, little y? Beautiful. 1/2 times 1/2 times 1/2. 1/8. What's the probability of getting big P, big P? Little y, little y? Little r, little r? This gives us 1/16. And our last one, little p, little p? Little y, little y? Little y, or little r, little r? Okay. Yeah. It's splitting up and doing each trait individually instead of doing all 3 at the same time. And then using the probabilities of the individual Punnett squares and multiplying them together to figure out the trihybrids. So this is step 2. We took all of our genotypes for our wanted phenotypes, and we found our probabilities. That's our multiplication rule. But we want to know what's the likelihood of getting any of these. So what do you think we're going to do with our end probabilities? What do you think we're going to do with these numbers in the square? Add them together. Just like we did with our single Punnett square to find out the probability that we got big R from dad and little r from mom, or big R from mom and little r from dad, right? We added those together to get 1/4. What we're going to do is we're going to use our addition rule. So our final responses, right? We use the addition rules of all of our different genotypes in step 3 to fulfill our condition. So 1/16, 1/16, 1/8, or 2/16. 1/16, 1/16, add them all together. There's a 6 in 16th chance of getting any of those phenotypes, or a 3 in 8th chance. Now we could fill out that trihybrid cross and go through and look for specifically all of the genotypes that would give us our right phenotype. Or we could do the math. Find our genotypes, do our multiplication rule, do our addition rule, and get 6 out of 16 instead of setting up the trihybrid cross, filling out the trihybrid cross, and multiplying or hunting and pecking for our correct answers. That makes sense? Okay. Yes, so another quick shortcut. We're going back to dihybrid crosses really, really quick, just because this is going to be important for you to know. If at any point in time you are asked a question that involves crossing 2 heterozygotes, right, and we want to know something about phenotypes, you never have to cross 2 heterozygotes in a Punnett square. Because our phenotypes will always be a 9:3 to 3:1 ratio. Bury 9:3 to 3:1 in your heads. This will definitely come in handy. 9 out of our 16 will have both dominant traits. 3 will have one dominant and one recessive. 3 will have the other dominant and the other recessive. And 1 will have both recessive. It does not matter. Does not matter what trait it is. It could be cattails. It could be plant color. It could be our pea pod shape. Doesn't matter what trait. At any point in time, if you cross 2 dihybrid heterozygotes, they have to be heterozygotes, you will always, always, always get a 9 to 3 to 3 to 1 ratio. So if I said, okay, we're crossing 2 heterozygotes with our initial dihybrid pea plant, whatever, and I said, how many will have both dominant traits? Without even thinking about it, you should say 9. Or maybe think about it a little bit. But without having to do the Punnett square, it'll always be 9 with both dominant traits. How many will have both recessive traits? 1. How many will be dominant for color but recessive for shape? 3. How many will be recessive for color but dominant for shape? 3. Always. Right here, right? We've got 1, 2, 3, 4, 5, 6, 7, 8, 9 with our 2 dominants. 3 with our first dominant and second recessive. 3 with our first recessive and second dominant, and 1 with both recessive. This 9 to 3 to 3 to 1 ratio will definitely come in handy. And then, yeah, there are practice problems on Canvas. It's just like kind of a lengthier exit ticket using these probabilities, answering some questions. So you have the time to do that. If you have any questions, please let me know. Then tomorrow we get to talk, I believe, about complex inheritance. Yeah, so codominance, incomplete dominance, all that fun stuff tomorrow. Cool beans? Cool beans.